Today we'll be talking about the properties and functions of glial cells in the central nervous system. And as always, write down and bring to class at least 2 questions about this recorded lecture, and we'll discuss the questions in class. There are several different types of glial cells in the central nervous system. The most prevalent type of glial cell is the astrocyte. A second type of glial cell, one that was discovered just within the last decade or so, is the NG2 positive cells. These are also called oligodendrocyte progenitor cells. Another important class of glial cells in the CNS are the oligodendrocytes. These, of course, are the myelin-forming cells of the CNS, and their homologs in the peripheral nervous system are the Schwann cells, making myelin around axons in peripheral nerves. And finally, there are the microglial cells of the CNS. Now today we're going to be concerned primarily with the functions of astrocytes, and astrocytes have many different types of functions. Among these are that they provide structural and metabolic support for neurons. These are the classical functions that astrocytes have been believed to subserved over the past century or more. It was assumed that these were the main functions of glial cells in the CNS, and indeed astrocytes do provide structural and metabolic support. Astrocytes also play a very important role during the development of the nervous system. They function as stem cells for neurons and form neuronal progenitor cells, and they also play an important role in guidance of migrating neurons. Both during development and in the adult brain, glial cells also provide trophic support for neurons. Another important function of astrocytes is the regulation of extracellular ion levels in the CNS, particularly potassium levels. They also play an important role in regulating extracellular concentrations of neurotransmitters by taking up neurotransmitters once the transmitters are released at synaptic terminals. And they also can release their own transmitters, gliotransmitters, and by doing this, glial cells can modulate synaptic transmission. And finally, by releasing vasoactive agents, glial cells can regulate blood flow in the CNS. One of the functions that has been classically attributed to astrocytes is the metabolic support of neurons. And there are several reasons for this. For one, we know that glucose is the main energy source for the brain, and there are stores of glucose in astrocytes in the form of glycogen granules. Glycogen is a polymer of glucose, and it's thought that in times of high energy demand, this glycogen can be broken down within astrocytes and the resulting glucose supplied to neurons. Second reason that astrocytes have been thought to provide energy to neurons is that the glucose, of course, originally comes from the blood, and blood vessels in the CNS are completely surrounded by the end-foot processes of astrocytes. So astrocytes are perfectly positioned to transport glucose from the blood to neurons. In addition to this function of supplying glucose to neurons, a new hypothesis has been offered in recent years, and this is called the lactate shuttle hypothesis. And this hypothesis holds that the metabolic breakdown of glucose into ATP occurs not only in neurons, but it occurs in two separate compartments. The first is in astrocytes, where glucose is broken down to lactate by the process of glycolysis, and 2 ATP molecules are made during this process, and then the lactate is transported from the astrocyte to the neuron, where lactate would enter the TCA cycle, and additional ATPs, up to 36 ATPs, could be made in the neuron. So This hypothesis holds that glucose breakdown occurs in these two separate cellular compartments, and there is evidence supporting this hypothesis. For one, there are transporters, both in glial cells and in neurons, that can transport lactate. In the astrocyte, there is the monocarboxylate transporter 1, MCT1, that transports lactate from the astrocyte into extracellular space. And then in neurons, they express a second MCT transporter, MCC2, which would transport lactate from the extracellular space into the neurons. And as I mentioned, once lactate is transported into the neuron, it can be converted to multiple ATPs through the action of the TCA cycle and oxidative phosphorylation. Now, I should mention that this method of supplying energy to neurons, the lactate shuttle hypothesis, is somewhat controversial. It is almost certainly true that this lactate shuttle process does occur, but what's not clear is what percent of the energy of neurons is derived from this. So what percent of energy comes from lactate, and what percent of energy comes from the direct supply of glucose in neurons and the breakdown of glucose. Another important function of astrocytes is the regulation of extracellular potassium levels. When neurons are activated and they depolarize, they release potassium. Neurons that generate synaptic potentials or action potentials depolarize by the influx of sodium, and then this is balanced, and the resting membrane potential of neurons is restored by potassium efflux. Now, normal extracellular potassium levels in the CNS are quite low. They're approximately 3 to 5 millimolar. So when potassium is released from active neurons, the extracellular potassium levels increase significantly, from 3 to 5 millimolar up to a ceiling level of about 12 millimolar for intense neuronal activity. We know that when extracellular potassium levels increase, this can in turn depolarize neurons and change their excitability. So in fact, there are very powerful mechanisms for regulating extracellular potassium levels so that the extracellular potassium never varies very much from a basal level of 3 to 5 millimolar. It turns out that glial cells and astrocytes in particular are the primary cells that regulate extracellular potassium, and this regulation occurs by several different mechanisms. When potassium is released from active neurons, the excess potassium that is dumped into extracellular space can be temporarily stored within astrocytes by potassium uptake, and this can occur by a passive influx of potassium and a counterion, typically chloride. So there can be an influx of potassium and chloride into astrocytes, and remember that astrocytes have a very high conductance to potassium. They have many potassium channels that are open at rest, so that this potassium conductance allows this passive influx of potassium into the astrocyte. Potassium can also be taken up into astrocytes by an active transport of the sodium-potassium ATPase, and this also occurs. A third method of potassium regulation in the CNS is by a process called potassium spatial buffering, and this is what's illustrated in the figure in this slide. When potassium is released from active neurons, it can enter a astrocyte by influx through the open potassium channels. This depolarizes the astrocyte, and this drives out potassium from distant regions of an astrocyte, or as is shown in this figure, the depolarization can actually be transmitted through gap junctions to distant astrocytes in a astrocyte syncytium, and then the potassium will be released from distant sites. The arrows, the blue arrows, show the resulting current flow with potassium and current entering the astrocyte in regions where potassium is raised, and then potassium is released from other regions of astrocytes where extracellular potassium is lower. The net result is that the excess potassium from active neurons is transported to distant regions of the CNS. Again, this process is called potassium spatial buffering and plays an important role in regulating extracellular potassium levels in the CNS. Glial cells play a very important role in the developing brain. In the developing brain, in very young brains, astrocytes are not found. Instead, there's a related type of glial cell called a radioglial cell. These are cells that extend all the way from the ventricle and deep in the brain, extending all the way up to the pial surface of the brain. It has been shown, and this was discovered just in recent years, that these radioglial cells divide regularly, and when they divide into asymmetrically, where one daughter cell is another radioglial cell, and the other daughter cell is either a neuron or a neuronal progenitor cell. And in fact, it's believed that most of the neurons in the cortex derive from these radioglial cells, which in fact are neuronal stem cells. Once neurons are born down by the subventricular zone, they migrate towards the pial surface of the brain, and these radioglial cells once again play a very important role in this process, as is shown by this right-hand picture. This is in a tissue culture showing a neuron crawling up and migrating along a radioglial cell. And indeed, the migration, the movement of new neurons or neuronal progenitor cells towards the pial surface of the brain occurs along these radioglial cells, and in fact, if this process is interrupted, for instance, by adding antibodies against glial surface molecules, this blocks the migration of neurons and disrupts the normal development of neurons in the brain.